We're going to move on to limb-girdle muscular dystrophy. I just want to introduce with a couple of thoughts. Our speakers today are Peter Anderson from Vita Therapeutics and Douglas Sproul from ML Bio. Let's see if this... Oh, there we go. So there's lots of puzzles still to solve in limb-girdle muscular dystrophy. And so this is just an example of some of the questions that people around the world are asking as we try to understand this disease better and come up with more therapies. So genetic subtype classification, as a lot of you know already, we used to call them limb-girdle type 1, limb-girdle type 2, and then use letters. And as you can imagine, we ran out of letters for limb-girdle type 2. So a few years ago, an expert panel convened and proposed LGMD-D for dominant and LGMD-R for recessive. And that system has been working well. I think because we'd been using the old system for several decades before that, it's been a tricky transition for people to think of using the new terminology, but I think it's becoming more widely adopted. And I think this will keep evolving as we have more information. There are also diagnostic dilemmas. Unfortunately, there are lots of cases of limb-girdle that are difficult to solve genetically. And then variant classification, as anyone who is in the clinic knows, we see a lot of variants of unknown significance in limb-girdle genes, and it can be quite frustrating both for the clinicians and the families and the patients. And then therapies, which is what our speakers will be talking about today, which are very exciting developments. How can we develop disease-modifying therapies for limb-girdle dystrophy? So our speakers today, as I mentioned, are Peter Anderson. He's going to talk about iPS-based cell therapy for limb-girdle muscular dystrophy type 2A R1, and Douglas Sproul will speak about preliminary results from MLB 01-003, an open-label phase 2 study of BBP 418 in patients with limb-girdle muscular dystrophy type 2I. And I do want to thank the speakers for coming today. And so I'll hand it off to Dr. Anderson.
Excellent. Okay, this is great. So I guess like from the title of these talks, I can see that we probably have a very basic biology talk and a more clinical oriented talk. So, but let me introduce myself. My name is Peter Anderson. I'm the current Chief Scientific Officer at Vita Therapeutics. And today, as you mentioned, we're going to talk about, or I'm going to be talking about how we are developing an autologous iPS-based therapy for limb-girdle muscular dystrophy 2A. This is good. So just briefly about Vita Therapeutics. We're a biotech company. We were founded in 2019. We focus on developing cell-based therapies for indication or patients with an unmet need. We have a number of preclinical programs in the pipeline. Today I'm going to be talking about our neuromuscular platform, and in particular VTA100, which is the program designed to restore muscle tissue in limb-girdle muscular dystrophy 2A. We were founded out of Johns Hopkins University, and as of today we've raised about $70 million, part in non-dilutive funding, and in 3 VC-led investment rounds. So, briefly about limb-girdle muscular dystrophy 2A. It's an autosomal recessive disorder that is characterized by muscle wasting in the hip and the shoulder areas. It's caused by patients' inability to produce functional CAPN3 protein due to mutation in the CAPN3 gene. And it's associated with abnormal sarcomere alignment and eventually remodeling, which in turn leads to this muscle loss and muscle weakness. So all of you here are very familiar, I would expect, in terms of muscle biology, muscle homeostasis, and the response to injury. So I'll just briefly summarize it because it's pretty important for this talk. So as you can see out here, which one is the... Is there an indicator? Nope. Oh, there's no laser on this one? Anyway. So as you can see out here to the left, satellite cells, they surround the muscle fibers, and upon activation by stimuli such as an injury or other type of response, these satellite cells, they're able to undergo a process called asymmetric cell division. They give rise to a daughter satellite cell and a myoblast. A myoblast is what we in biology refer to as a transient amplifying cell. These can expand rapidly in numbers, and then they can fuse with one another and form de novo muscle fibers, or they can fuse with the injured muscle myofibers to repair them. So later on, here about 15 years ago, the discovery of iPS technology, which is technology that allows the generation of pluripotent stem cells from patients through a process called cell reprogramming. It really excited a lot of us due to the potential of generating potential personalized tissue cells to be used for either disease modeling, for drug discovery, and even for cell replacement therapy, which I'm going to be talking about today. The excitement was obviously very clear because you could potentially avoid the immune rejection that is typically associated with allogeneic cell transplantation. So when this discovery was made, there was a number of labs around the world that started working on these differentiation efforts to generate all types of tissue cells using various protocols to differentiate all types of cells from these iPS cells. I started at this time my lab at Hopkins, and I started to collaborate with two investigators, one called Catherine Wagner, which many of you are probably familiar with, another Gap-Sang Lee, who were both together in a collaborative effort working on developing a method to differentiate these iPS cells into muscle tissue. They eventually succeeded, and they used this technology to demonstrate how it could be used to basically study Duchenne's muscular dystrophy in the lab or in a dish. So we started to collaborate. We published some papers together, both to where we demonstrated how we could study muscle biology in a dish and also be able to discover new drugs using these kind of models. But about a year ago, we published this last paper, as you see down here, where we studied the regenerative aspect of these cells and saw what they could do once we transplanted them into mouse models. So from that paper, I just want to highlight two important figures. The first one is demonstrating how we could take a mouse, we could injure the TA muscle or the tibialis anterior, and then we can inject these progenitors that we have developed from these iPS cells or pluripotent stem cells. And what we saw about 4 weeks later, as you can see here in white, is that they basically entered the muscle tissue and started to regenerate all this damaged muscle fibers. Along with that, we were able to also see that a number or subset of these cells, they engrafted in the basal laminar and still expressing PAX7, which is a marker of satellite cells or early progenitors, and it led us to think that these cells were actually engrafting as satellite cells. And should they do so, they should also hold a long-term regenerative potential. And this is exactly what we see here in the next experiment below, where we actually see that when we had injected these cells and we then re-injured these mice, we saw that after 4 weeks, the muscle kept on regenerating, indicating that our satellite cell or our muscle-derived cells were actually repairing the injured muscle tissue. So, as for Vita Therapeutics, as I mentioned, this technology was adopted by Vita and is the foundation of Vita's lead program called VTA100. This is an autologous program, and it's designed to regenerate muscle mass in limb-girdle muscular dystrophy 2A patients. All preclinical partnerships has been established, and we expect to file an IND next year. Let's see. So the program is involved. There's 4 steps to the program, and basically what we see here is what we're doing. We're taking a blood sample, we reprogram into iPS cells. We're then using CRISPR technology to repair the CAPN3 mutation. We differentiate and we then isolate these progenitor cells, and we can then expand them afterwards. Now, once we have these cells, then they have to undergo a number of QC assays. I've listed a few of them here, but it's not limited to these. But some of these assays involve sterility and viability, as you can see here. But we also examine the cell residuals from these cells, whether there's any iPS cells left, but also we look at cellular identity. We do that by flow cytometry. We evaluate the cell's ability to form muscle fibers and to be able to fuse, so we can actually get like an idea how good these cells they are in a dish. Of course, we monitor CAPN expression, which we have actually corrected. And then finally, we're doing whole genome sequencing and karyotyping to make sure that these cells are actually still intact and that they should be functional as expected. So after that, we continuously test them, their ability to regenerate and repair muscle tissue in a number of injury and genetic in vivo models. Due to the time restrictions here, I don't have time to go over all these different things, so I'm just going to show you some key data points that we have from these studies. So the first evaluation that we do is 24 hours after we have injected these cells. Here we look at the biodistribution of these cells. I'm not showing any images of that here, but 4 weeks after, that's where we start to look at the regenerative potential. And what you can see here on the images out here is, again, the formation of fibers. You can see that here in green, where we have from the transferred section, we have the engraftment of these cells. They are regenerating the injured muscle tissue. And we also do a lot of single cell sequencing to examine the state and maturation of these cells as well. 3 months is typically the next analysis that we do. And this is actually pretty important because this is where we kind of look at the ongoing regeneration. And this is some of the stuff that really excites us. So as you can see here on this bar graph, we can see we have no injury at 1 month and 3 months. And when we inject these cells, we can see that there's really no effect over time. We wouldn't expect that because it's an uninjured muscle, so we wouldn't expect a lot of contribution from our cells. However, when we do this in the injury model, we actually see a big difference from 1 month to 3 months, demonstrating that these cells, they continue to repair and replace the muscle tissue. And finally, we look at 9 months. We can see that our cells, they persist. This persistence is giving a lot of confidence that these cells are actually be able to persist and provide a long-term cure or effect on these patients. So just to summarize, we've developed a protocol for producing autologous iPS cells at a clinical scale. We have demonstrated consistent and reproducible production of these myogenic progenitor cells from multiple donors. And in the preclinical studies that I just briefly showed you here, we have demonstrated both activity and long-term persistence of these transplanted progenitor cells. Now, we expect to file an IND later this year, and to commence the studies later this year and file an IND in Q3 next year. So with that, that's it. Oh, and we actually do have one of our principal scientists, Sonny, here, who's at poster 269. So if you have questions around this, you can go to that poster and you can ask all the complicated questions. If you. Thank you.
Thank you very much. And we will be taking questions at the end of the session. And Dr. Spruill is next.
All right. So see if we can get it up there. Perfect. So my name is Doug Spruill. I'm the Chief Medical Officer at ML Biosolutions. I'm also a pediatric neuromuscular specialist back in the old days before I came to the dark side. And you know, I can't remember when the last time, or the first meeting I came to, I think it was 2008, 2009, and just to see the transformation of this meeting and our field is really awesome. And it's fantastic to see, and it's a privilege to be a small part of it. I'm going to present our preliminary results from our phase 2 study of BBP-418 in patients with limb-girdle muscular dystrophy type 2I. It's also in the new nomenclature R9 FKRP related. So my disclosure, ooh, this is tricky. There we go. Disclosure, I am Chief Medical Officer at ML Bio, so take every word I say as being biased and agenda-laden and judge it accordingly. BBP-418 has not been approved to treat patients by any regulatory authority in any country, and our studies are ongoing. So where do you point it at?
You point this way.
Cool. So where do you point it? Oh, if you put it on the... Ah, there we go. I'll just stop playing with it. All right, so very good. So alpha-dystroglycan, or ADG, is the core glycoprotein impact in this disease. Dysfunctional FKRP enzyme activity results in impaired glycosylation of alpha-dystroglycan. This disrupts the function of this critical muscle-stabilizing protein. And so for this reason, LGMD2I is one of which results from a biallelic mutation of FKRP, is considered one of a number of diseases that are kind of lumped together as dystroglycanopathies. So there we go. All right. So BBP-418 is a synthesized version of endogenous ribitol, and so it's provided in a powdered form, dissolved in water, and consumed orally. And on the left side of this figure, we see what happens in the untreated disease state. So we have partial loss of function of FKRP, and this results in widespread dysfunctional hypoglycosylation of alpha-dystroglycan in myocytes. And this results in insufficient full-length functional alpha-dystroglycan, which leads to destabilization of the muscle membrane and increases susceptibility to injury and the ultimate development of a muscular dystrophy. And on the right side, we see the proposed therapeutic mechanism. And so while the FKRP enzyme doesn't work correctly, it works well enough that by providing supraphysiological levels of substrate, you can harness that residual enzyme activity and drive forward full-length alpha-dystroglycan glycosylation. So it's a simple but elegant solution to obviously a complex problem. I wonder if it's a battery issue. Dab nabbit. It's going to go twice one of these times. So, all right, perfect. So we developed a novel bioassay to measure the glycosylation of alpha-dystroglycan in tibialis anterior muscle biopsy samples. And what we know from the literature, published natural histories and the established kind of paradigm disease, is that patients who are homozygous for the L276I mutation, some people call it the common mutation, Dr. Matthews calls it the Viking mutation, tend to have somewhat later onset, less severe progression of disease compared to patients who have other genetic profiles, typically one L276I mutation and one other more severe mutation. And as we would expect, when we look at this bioassay in natural history study patients, alpha-dystroglycan glycosylation is reduced in these populations, mirroring what we would expect in the clinical severity of the disease. So on the left side, we see measurement of glycosylated ADG as percentage of levels seen in healthy donor tissues in the tibialis anterior. And so what we see is that the L276I homozygotes are approximately 12 to 13% normal, whereas patients with other more severe genotypes tend to are hovering around 5% of the amount of glycosylated alpha-dystroglycan as compared with healthy tissues. On the right side, we see the stability when looking at baseline versus 6, 9, or 12 months longitudinally, and we see stability in the assay, at least over short periods of time that it would be commensurate with clinical studies. On the left pair is the L276I homozygotes, the right pair is patients with other genotypes. So the MLBio or MLB-01003 study is an open label, single site dose escalation phase 2 study that enrolled 14 patients. So in addition to safety, we looked at clinical measures including the NSAD, the 100-meter time test, 10-meter walk, PUL 2.0, FVC, and a number of other measures longitudinally in the course of this ongoing study. Let's try. Everybody else seemed to figure it out. It must be me.
That has a pointer function.
And then it did what there we go. All right, so to this point, BBP-418 has been well tolerated in patients with only minor GI-related adverse events to date. We've seen 14 possibly or probably related adverse events, including diarrhea, dehydration, nausea, vomiting, dyspepsia, gastroenteritis, and headaches. We've had no discontinuations or interruptions in therapy. The 3 severe adverse events were all unrelated to therapy. So, come on, give me some love. There we go, perfect. So, and obviously this is a small open label study. Take the results for what it's worth. But on the left side we see the increased glycosylation of alpha-dystroglycan following administration of BBP-418. So in forest green are the plots of the L276I homozygotes, and you see an increase from approximately 17% of normal to approximately 40% of normal sustained over periods of time up to 15 months. On the right side, we see an almost doubling as well, going from around 6 to around 10% of normal expression following administration of BBP-418. On the right side, we see CK. So creatine kinase, obviously, it's imperfect but widely used marker of muscle breakdown, and we see a decrease of more than 75% that's from baseline that's sustained over periods of time of up to 15 months. We're obviously really excited. Again, this is an open label study. It's small, but we're starting to see emergence of some clinical signal as well. The red dashed lines reflect patients in the phase 2 study treated with BBP-418. The blue lines are patients who have been seen as part of the MLBio-sponsored natural history study. So what we are seeing at this point is some modest improvements in 10-meter walk test velocity, the time to complete the 100-meter time test, and NSAD score when compared with their baseline. And obviously this contrasts with the decline that we see over time in natural history patients, which is seen in the blue. So this sets, to kind of summarize, we're seeing increased glycosylation of alpha-dystroglycan increased glycosylation of alpha-dystroglycan following treatment with BBP-418. We're also seeing a large and sustained reduction in CK and some early clinical signals. And this is in the context of a reassuring safety profile to this point. Obviously, it's very early with no treatment-related SAEs or dose-limiting toxicity. And obviously, we're building upon that. We're extremely excited to be moving over the course of the next few months, several months, into phase 3 study of BBP-418 in LGMD2I. It's going to be a multi-center, multinational, randomized, double-blind, controlled trial. We're going to be enrolling approximately 80 to 100 patients, and with broad criteria, including both L276I homozygous and patients with other genetic profiles, and children at this point 12 years and older, as well as adults. So it's going to be a pretty broad study. We're very excited to be moving into it. The NSAD is going to be our primary endpoint, and we're aiming to start later this year. So with that, I want to thank my colleagues, and I probably run over time, my colleagues, but also particularly the team at VCU, Amy Harper, Ruby Langesley, particularly patients and families. And we have two posters that are right next to each other, right next to the cocktail bar, 140 and 139. So I encourage everybody to stop by and talk to us more. So.